Genetic recombination is a process where genetic materials contained in two separate genomes are brought together within one unit. In bacteria, the recombination takes place by transduction, conjugation, and transformation. In this video, we are going to discuss how bacterial conjugation works. What is bacterial conjugation? Bacterial conjugation is the transfer of genetic material between bacterial cells by cell-to-cell contact or by a bridge-like connection between two cells. How does it happen? Bacteria can transfer plasmids through conjugation. Plasmids are small DNA molecules within a cell that are physically separated from chromosomal DNA and can replicate independently. They are most commonly found in bacteria as small circular double-stranded DNA molecules. A donor cell extends one or more projections, the pili, that attach to a recipient cell and pull the two bacteria together. Here we have a donor cell with a sex pilus slowly attaching to a recipient cell. For bacterial conjugation, we have the conjugative plasmids or the fertility plasmids, the F factor. An F plasmid can exist as an independent unit or it can integrate itself into chromosomal DNA. These plasmids allow the transferring of copies of themselves to other bacteria. This is made possible because other than the genes responsible for antibiotic resistance, the F factor also contains information for the formation of the sex pilus and insertion sequences that assist in plasmid integration. The sex pilus enables conjugation and is responsible for the direct cell-to-cell contact. Here we have an F plus cell. An F plus cell is a bacterial which contains conjugative plasmids or the F factor. This allows the F plus cell to connect with an F minus cell, which does not contain a fertility plasmid, through the sex pilus. The sex pilus from the F plus cell is projected and makes contact with the F minus cell. The sex pilus then contracts, bringing the two cells together, which now creates the type 4 secretion system. To begin the transfer, the F factor is cut in the origin of replication by the protein assembly called the relaxosome. The relaxosome is the complex of proteins that facilitates plasmids during bacterial conjugation. The relaxosome makes a cut from which the tDNA strand is separated. Accessory proteins from the relaxosome are released, but a portion of the relaxosome, called the relaxase, remains attached to the tDNA. The tDNA relaxase complex is then recognized by the coupling factor and transferred to the secretion system. The secretion system then pumps the tDNA relaxase complex into the recipient cell or the F minus cell. The F factor of the donor cell was also replicated to be double-stranded during the transfer of the tDNA molecule to the recipient cell. Once the entire tDNA molecule is transferred, the relaxase joins the ends to make a circular DNA structure. Now it is an F plus cell that can go on and conjugate with other bacterial cells. Earlier, we talked about how F plasmids can exist either as an independent unit or can integrate itself into a chromosomal DNA. The F plasmid also contains the insertion sequences which help in assisting plasmid integration. And when this integration happens, the derivative of F plus cells are now called HFR cells, or the high frequency of recombination cells. When the sex pilus of the HFR cell comes in contact with the F cell, F minus cell, or the recipient cell, it then contracts, pulling the two cells together, creating the type 4 secretion system. One DNA strand is then cut from the F origin while the chromosome is replicated. The donor strand is also being replicated as it enters the recipient cell. When the two cells separate, the donor cell chromosome is restored. The recipient cell integration will lead to recombination of gene alleles. Linear DNA is eventually degraded. A portion of the donor chromosome DNA also gets transferred into recipient cells because of the F plasmid being integrated onto the donor chromosomal DNA. When integration between an F factor and a chromosomal DNA happens, it can also disintegrate itself back from the host chromosome after the formation of an HFR cell. Sometimes, disintegration may occur incorrectly. When this happens, the F plasmid may bring some of the host genes along with it from its disintegration sites. Now this creates the F prime cell. And just like how the F plus cells and the HFR cells conjugate, F prime cells begin by a connecting sex pilus, creating a type 4 secretion system. The F prime plasmid begins replication and transfer through the secretion system into the recipient cell. The F prime plasmid is now replicated and transferred. Again, Why is bacterial conjugation important, and what is its purpose? Bacterial conjugation is the ability for one bacteria to transfer genetic material to another via a physical bridge between the cells. Conjugation is used in nature to share beneficial genetic material between bacteria, such as antibiotic resistance, which is very important in cell survival.